A very good morning to all of you. So today discuss an important topic, that is Crispr Cas9. Crispr Cas9 is one of the important topic for molecular biology as well as for the recent advances for MD exam and also other competitive exam also. Okay. So Crispr Cas9. So what is Crispr? Crispr Cas9 is nothing but it is a gene editing tool. It is a tool which is used for the gene editing. Although a number of tools used before the discovery of Crispr Cas9, like the most important, that is in recombinant DNA technology, there is use of restriction endonuclease and there is a zinc finger nucleases technology also. But these older technique having a lot of disadvantages because of their expensiveness, and it is not easy to prepare all these genetic gene editing tools. So nowadays with the advent of this Crispr Cas9, it helps a scientist a lot in the field of your treatment of genetic diseases and in research purposes. So it is nowadays very important gene editing tool which is used for editing a variety of gene. Okay, so this is the use of the Crispr Cas9. So today we will understand how this Crispr Cas9 works. In next session we will understand the advantages and application. Okay. So CRISPR, first understand what is the full form of this Crispr Cas9. CRISPR C stand for cluster, R for regularly, I for interspace, S for short, P for palindromic, R for repeats, and Cas9 is a protein. So this is nothing but cluster, regularly, interspace, short, palindromic, repeats. Okay. Now understand how this CRISPR has been discovered by the scientists. So this Crispr Cas9 is nothing but it is a defensive mechanism in bacteria against the viruses. Okay. So this is the defense mechanism which is present inside the bacterial cell, and it helps the bacterial cell to protect the bacteria against the viruses. Okay, so basically it is a defense mechanism. Now how it works inside the bacterial cell we will understand. So this blue one is what? This is the diagrammatic representation of your bacterial cell, and this blue one is the bacterial cell wall. Okay, as you know, the genome of bacteria is circular in nature, so this is the circular genome of the bacteria, and this circular genome of bacteria having this CRISPR gene. Okay, inside the circular genome of bacteria there is a CRISPR gene, and this CRISPR gene consists of, there is a clustering. This black one is what? There is a cluster of this is cluster regularly. So it is a regularly cluster. Okay, regularly cluster that cluster are interspaced by this green spacer. Okay. These black one is what? These black one is the clustering of bacterial DNA, and these are regularly present, and these are the short palindromic repeats. Understand? So what is this? This black one, this black triangle is the clustering of bacterial DNA, and these are present inside the bacterial genome in the repeated sequence, and these are the palindromic repeats, and these palindromic short sequences are interspaced, or these are the interspaced by this spacer DNA. And this green one is your spacer DNA, and this spacer DNA is nothing but it is a part of viral DNA. Understand it again. This CRISPR gene consists of a black triangle, that is of bacterial DNA, and this bacterial DNA are present in cluster form, and it comes at a regular interval, and these regular interval are like break or interspaced by the spacer DNA, and this spacer DNA is nothing but this green one. This green spacer DNA is the viral DNA. Okay, and what now? This whole unit is known as CRISPR gene. What do you understand by palindrome? Palindrome means whether you can read from this right side or left side, it is similar. Like dalda, madam, all these what? These are the palindromic repeats. Suppose in one strand of DNA there is sequence GAATC, and another strand 5' GAATC. So this is what? Palindrome. Okay. When this palindrome will undergo the transcription, that is when from this palindromic sequence there is a formation of RNA, then it will form a hair loop like structure, or you can say hairpin like structure. This is what? This is hair loop like structure. Alright. Now let's understand, beside in cluster genome, in this circular genome of bacteria, beside this CRISPR gene, is circular genome also consists of this Cas gene. Okay. in circular genome of bacteria, one is there is a CRISPR gene, and beside this CRISPR gene there is a Cas gene, and this Cas gene will form the Cas9 protein. Not only Cas9, although Cas gene will form number of Cas protein like 1, 2, 3, 9, 30, so on, so on. Not only Cas9, but we have taken consideration of Cas9 protein. Why? Because this Cas9 protein has the property to cut the double-stranded DNA. Okay, because this Cas9 has the property to cut the double-stranded DNA. Similarly, there is a Cas13 protein which can cut single-stranded RNA. Okay, so now you can easily understand why in this CRISPR we have taken only Cas9 protein, because these Cas9 protein are capable to cut the double-stranded DNA. And for the gene editing, if we are using this CRISPR as a gene editing, so our interest of targeted interest is double-stranded DNA we have to cut, okay, because eukaryotes having double-stranded DNA. Alright, so that's why in this technology we have taken only Cas9 protein, although bacteria from Cas gene synthesize a variety of Cas protein. Okay, so from this CRISPR gene there is a RNA will form, and from this Cas gene there is a synthesis of this Cas9 protein. Okay, besides this, circular bacterial genome also consists of another gene, that is your target gene, and when this target gene will undergo transcription process, then it will form the target RNA. Clear? Now comes what will happen. So from this CRISPR gene, what will happen? This RNA assembly will form with the help of enzyme RNA polymerase. Okay, so this green one will form your spacer RNA. From this spacer viral DNA, it will form spacer RNA, and it is around 20 nucleotide sequence. And from this black one of triangle bacterial DNA, it will form CRISPR RNA. Okay, and from this red one of target gene, it will form your target RNA. So this is your whole assembly of CRISPR RNA, which consists of green one spacer. It is a part of your viral DNA. From triangle of black bacterial DNA, it will form CRISPR RNA, and from this target gene there is a target RNA. Now this CRISPR RNA and this target RNA together, they are known as guide RNA. Why it is known as guide, we will understand later. Okay, so this is the assembly. Now suppose if this is the virus which will infect the bacteria, and the virus which will infect the bacteria is known as bacteriophage. Okay, so when this bacteriophage will infect this bacterial cell, it will leave its genetic material inside the bacterial cell. Okay, this bacteriophage consists the DNA, and it will leave its DNA inside the bacterial cell. As soon as it will leave its genetic material, bacteria immediately recognize it. Okay, because if it recognize it and see it, it's a foreign material and we have to kill it. Okay, so what will happen? As soon as it will enter and bacteria recognize it, immediately this Cas9 protein will form assembly with this whole RNA part. So there is a next thing what happened. This red one is your Cas9 protein, and it will assemble with this whole RNA unit. Okay, now what will happen? This guide RNA, and this guide RNA what? This guide RNA is a combination of your CRISPR RNA and your target RNA. So what is the function of this guide RNA? This guide RNA will guide this Cas9 protein to find out the specific complementary sequence in the viral DNA which is complementary to this spacer RNA. Understand? So once again, what is the function of this guide RNA? This guide RNA will help this Cas9 protein to recognize the specific target sequence on viral DNA. That sequence is complementary to the spacer sequence of the viral DNA. Okay. So once this guide RNA will recognize it, then what will happen? Then this Cas9 protein will make a cut like a scissor on that recognition site. Okay, this is the, suppose this is the complementary sequence. This sequence is the complementary to this spacer RNA, and guide RNA will recognize it, and then what is the function of this Cas9 protein? It will act as a scissor and it will cut that viral DNA, and ultimately what will happen? The viral DNA is cut. And once viral DNA is cut, it cannot hijack the machinery of bacteria and it cannot infect the bacteria. So by this mechanism, bacteria will protect or defend itself against the viruses. Okay. Now understand how this spacer sequence comes inside this CRISPR gene. Suppose this is the same bacteriophage This is the same bacteriophage which infect the bacteria first time. Okay, before this bacteriophage, this same bacteriophage infect the bacteria for the first time. So as soon as it infect the bacteria first time, so it will take some time to develop the defense mechanism against it, because you know, first time if something happened or foreign material will infect, then immune system will take some time. Similarly happened in bacteria. So this is the bacteriophage which will infect the bacteria, and it will leave its DNA inside the bacterial cell. This green one is the viral DNA, and this viral DNA consists of T sequence. It have one protospacer sequence, and above this protospacer sequence there is a PAM. What is PAM? This PAM is your protospacer adjacent motif, and this PAM sequence is present above this protospacer sequence. As this viral DNA enters, then what will happen? There are Cas1 and Cas2 protein, and these Cas1 and Cas2 protein will recognize. What is the function of this Cas1 and Cas2? This Cas1 and Cas2 will recognize this PAM sequence, okay, which is present above the protospacer region. So it will recognize this PAM sequence and get attached to PAM sequence, and it will form a cut at the PAM sequence. And when this will cut, then what will happen? This protospacer, then what will happen? This protospacer, this one, will release, and this released protospacer will get attached to this bacterial genome, circular genome, and it is a part of the Crispr gene. And what is the function of this protospacer spacer? It will help the bacteria so that when again and again next time same bacteriophage enter inside the bacteria, it will help bacteria to cut the viral DNA, and it will protect the bacteria against the viruses. Okay, so this is the mechanism how easily the Crispr act inside the bacteria, and with the help of this Crispr Cas9 protein, how bacteria protect itself against the bacteriophage. So by this studying this defense mechanism, scientists make an idea ki if bacteria can cut a sequence of viruses at a specific position, so by using this technique we can also cut the DNA of our interest, and when we can use it as a gene editing tool. Okay, so by this technique, in next session we will understand what are the application and what are the advantages of CRISPR. So at this level you should know what is CRISPR, what is the use of CRISPR, and how it works inside the bacteria. Okay, so I hope you like the video and you will share with your friends. So next video we will understand the advantages and application. Thank you.